Hello students, today we will study about the formation of thyroid hormones. We studied about the structure. We will see some points related to the blood supply and how the hormones are formed and why is thyroid gland different from other endocrine glands. The blood supply of thyroid gland. The arterial supply of thyroid is very rich as in other endocrine glands, and it is received from external carotid artery and from the thyrocervical trunk. Nerve supply. The 10th cranial nerve is vagus, and it has a branch called as the recurrent laryngeal nerve. This recurrent laryngeal nerve supplies thyroid also. We have already studied that the thyroid gland secretes two types of hormones. The follicular cells form T3 and T4, and the parafollicular or the C cells form calcitonin. We will study today about T3 and T4, triiodothyronine and T4 thyroxine. We have studied that T3 is the more active form and thyroxine is the less active form. Bulk of T3, the more active form, is derived by deiodination of T4 in the peripheral tissues, especially liver and kidney. So deiodinization of T4 does not occur in the thyroid gland. It occurs in the peripheral tissues and mainly in liver and kidney. One important question which is asked in exams is the functional and the structural unit of thyroid glands. The functional and the structural unit of thyroid gland are follicles, which are made up of follicular cells. See, thyroid, we have already studied, is encased by a thin connective tissue, which forms a capsule around this endocrine gland. Now, this capsule forms projections inside the substance of the lobes. These projections of the capsule further subdivide the gland into irregular lobular units. So the gland has many lobules, and these lobules are formed by the connective tissue capsule which enters inside the tissue of the gland. Now the lobules contain the cluster of follicles, and these follicles are the structural and functional units of thyroid gland. The follicles are filled with colloid. We have to remember that follicles are filled with colloid, and this colloid contains thyroglobulin. This is very important. Thyroglobulin glycoprotein. This thyroglobulin is secreted by the follicular cells. Rough endoplasmic reticulum of the follicular cells secrete this colloid. The degree of activity of follicle can also be assessed. When we cut histological sections, we can assess the activity of follicle based on the amount and appearance of colloid it contains. If the follicular lumina are larger, colloid is abundant and appears solid, the follicles are inactive. And if the follicles are active, the lumina are smaller and there is no little or no colloid presence. So if the colloid is larger in amount, the follicle is less active or inactive. And if the follicles are large, it means, and if the follicles are small and the colloid is less, it means that the follicles are highly active. As we can see in this diagram, see, the follicular cells, they are cuboidal in appearance, and inside these follicular cells, colloid is present. This one round structure is one follicle. So we can see three follicles. And between the follicles, we can see the parafollicular cells and the blood vessels capillary. All endocrine glands are highly vascular. So thyroid gland is highly vascular endocrine gland. Now, this formation of hormones, how iodine is used and how thyroglobulin forms the hormone. There are various steps involved in this. See, the first step is iodine trapping. Iodine trapping, binding of thyroid stimulating hormone, which is released by anterior pituitary. So TSH from anterior pituitary travels through the blood, reaches to the thyroid gland, then reaches to the follicle cells. So binding of TSH to its receptors in the follicle cells of the thyroid gland causes the cells to actively transport iodide ions across their cell membrane. So the trigger is binding of TSH. As soon as TSH binds, the sodium iodide symporter transfers the iodide ions from the bloodstream inside the follicle cell. So, see, this is a structure of a follicle, and this is the basement membrane, this is the cell, and the blood vessels are present outside the basement membrane. So, the TSH comes from blood and attaches to the TSH receptor on the follicular cell. As soon as TSH binds to the TSH receptor, the sodium iodide symporter, you can see in this diagram, sodium iodide symporter is which transport both the ions in the same direction, symport. So sodium and iodine, they both are transferred inside the cytosol of follicular cell. So iodine comes. As a result, the concentration of iodide ions trapped in the follicular cell is many times higher than the concentration of iodide in bloodstream. Daily requirement of iodine is 100 to 200 mu G. These are some values which we need to remember for competitive exams. Then we also need to remember some anti-thyroid agents. So the thiocyanate and perchlorate, these are anti-thyroid agents They limit uptake of iodine by inhibiting sodium iodide symporters, which are also called as the iodine pumps. See, in this diagram, this is the capillary. This is the capillary. Capillary is made up of squamous thin cells. So these are the squamous cells. Inside the capillary lumen, blood is flowing. These are iodide ions. And this blue line represents the follicular cells. So this blue part is the follicular cell. This yellow part is the colloid, which is present inside the follicle. So if you see, when TSH arrives, when thyroid stimulating hormone arrives and it attaches to the capillary, to the basal part of to the follicular cell, the sodium iodine symporter, Na plus I plus symporter, this is the protein. Sodium iodide symporter will import the iodide ions from the blood into the lumen of follicular cell. So the iodide ions are transferred inside the lumen. This is one more protein called as pendrin. If you see, we will study about pendrin later, but we have to remember that iodide is transferred from the blood into the lumen through sodium iodide symporter, which is also called as the iodine pump. And if sodium is not present, this pump will not transport iodide into the lumens. So symporter, both ions are necessary. When both ions are present, only then this transport will occur. So this is the first step in which iodine is trapped. Iodine is trapped from the blood into the follicular cells, and this iodine trapping is very important. If the blood has low level of iodide, if the diet does not have proper iodine and the iodide level in the blood is low, then this first step will be affected and all the other future steps will be affected, and the production of T3 and T4 will be affected. So this is the first step in the formation of thyroid hormones. The second step is we will study in the next lecture.